Good morning students, I am Shomanjit sir. Today I am here to deliver my lecture on topic cell membrane or plasma membrane. So this is a very important topic. Watch the entire video lecture to get basics out of it. So without any delay, let's start with the topic cell membrane or plasma membrane from the chapter cell. So let's start. So students, let's start with the topic cell membrane. This is from the chapter cell. So let's start. Plasma membrane was discovered by Nageli and Kramer in 1855 and its name was given by Pauli in 1931. What is this all about? It is a very thin flexible membrane and it allows nutrient into the cell and allows waste to leave the cell. Now plasma membrane is also called selectively permeable membrane. That means it selects which nutrient will go inside and which will go outside of a cell. It selects. So selectively permeable means the membrane controls the movement of substances in and out of a cell. That means which substances will come inside the cell and which will go outside the cell. Next, plasma membrane function is to allow the nutrients entering the cell and leave the cell. Now see, there is a fluid mosaic model. So you see this is a phospholipid bilayer and they have said, Singer and Nicholson said that in a sea of lipids, proteins are scattering. That means proteins are surrounding. That means in an entire ocean of lipid, proteins are scattered throughout. So there are two different types of protein. It can be extrinsic protein, it can be intrinsic protein. Extrinsic protein means those proteins that are present on the outer and inner, upper and lower surface of the plasma membrane. Clear? Outer and lower surface. And those are intrinsic, will be present internally. Clear? So you see in a phospholipid chain, that circular blue color you have seen, round circular blue color, those are phospholipids. Clear? And top portion you see, top portion is a polar head and the middle portion, that means between one layer and another layer, there is a region given non-polar head, that is a tail. So top portion is the head and middle portion is the tail region. Clear? So middle portion is hydrophobic in nature, that means water fearing, and top portions are water loving, that means hydrophilic. So a phospholipid molecule consists of what? A glycerol backbone, two fatty acid chain and a phosphate group. So in the fluid mosaic model you see other components are also there like proteins, cholesterol and carbohydrate. So as I have said in the fluid mosaic model, in the entire sea of lipids, proteins are present somewhere. So if there can be a glycoprotein also, there can be glycolipid also. If glycogen is attached with lipid, this is called glycolipid. If glycogen is attached to protein, is called glycoprotein. Now see, proteins, what is the function of proteins? It transmit signal inside the cell. It acts as support structure. It provides pathway for substances to enter and leave the cell. So you see in a figure is given inside the cell, outside the cell, where are carbon chains. Clear? Which one is polar head, which one is non-polar head, where cholesterols are present. See, what is the function of cholesterol? It prevents fatty acid tail from sticking one another. Clear? I have said the inside portion is the hydrophobic, so it will not stick one with the another. That's the function of cholesterol is it will not allow them to stick one with the another. Now the function of carbohydrate, identify the chemical signals. Now what is passive transport? The movement of particles across the membrane without the use of energy. That means from lower concentration to higher concentration it will go. Clear? That means no energy will be used. Like for diffusion it will go for higher concentration to lower concentration. Here no energy will be used. So there are three modes of passive transport. One is diffusion, facilitated diffusion and osmosis. That means wherever no energy is required, it will go towards the concentration. Never it will go against the concentration. So whenever we go towards the concentration, no energy is required. That is a passive transport. So for diffusion, it will go from high concentration to lower concentration. So what is diffusion? It is net movement of water molecules from a region of higher concentration to the lower concentration. You see three figures are given there. Three jar is given there. See, uh, like if I take a jar filled with water and if I put like any dye, so what we see that it gets spreaded in throughout the beaker, isn't it? After some times, that is a diffusion in liquid. Diffusion can also be in diffusion in gases like incense stick or perfume. If I sprayed in the room, if I spray the perfume or incense stick, the smell will or the perfume will be scattered from here, from one room to another room, isn't it? So this is a diffusion. Now diffusion is controlled by temperature, pressure and concentration. Understood? So diffusion will keep on, or that means diffusion will do its work until the equilibrium is reached. Clear? Now diffusion in a cell, you see, from outside cell to inside, and center is a plasma membrane. So plasma membrane is a selectively permeable membrane. Clear? So you see the pink colored dye that I have given, it is entering inside the cell through a plasma membrane. Now what is facilitated diffusion? Movement of material across the plasma membrane using protein. Clear? Through protein it comes. See, there are two different types of proteins they have given: channel proteins and carrier proteins. Channel proteins are always present in the plasma membrane. They help to pass. But carrier protein means those molecules that will not pass, the carrier will help them to pass inside the cell, from outside to inside. Now what is osmosis? See, osmosis definition is what? See, movement of water molecules from a region of higher water concentration to the region of lower water concentration through a semipermeable membrane. Clear? Higher water concentration, that means what? Low concentration, to lower water concentration, that means higher concentration. Understood? So in other words, I can define the osmosis as it is a movement of water molecule from a region of lower concentration to higher concentration through a semipermeable membrane, or in your book another way of definition it has been given, it is a movement of water molecule from a region of higher water concentration to the region of lower water concentration. Here higher water concentration means what? Low concentration, and lower water concentration means high concentration. Clear? So there are three types of solution. One is isotonic, hypotonic and hypertonic. Isotonic solution means what? Equilibrium state. That means when water and dissolved substances are diffused in and out of the cell at the same rate. Clear? Whenever, if a cell is placed in a water medium and no water is getting inside the cell and outside the cell, that means it is an equilibrium state. The cell will neither shrink nor expand. This is isotonic solution. Now in case of hypotonic solution, hypo, clear? So cell will increase in size. So you see solute concentration is higher inside the cell. Understood? So if I place a cell in a water body, the cell will have higher water concentration. That means what? Water will have low concentration. So from low to high it goes. So water gets inside the cell, the size of the cell is increased. Reverse case is seen in hypertonic solution. Hypertonic solution means what? If I place a cell in a beaker, the water present in the beaker have a higher concentration as that of the cell. Clear? That means what? All the water, all the nutrients that are present inside the cell will come out. The cell will shrink. The diagram is given here, see? Shrinkage. So this condition is also called plasmolysis, and the reverse process of it is called deplasmolysis. Clear? That means whenever a plasmolyzed cell, that means the shrinked cell means what? It has been shrinked. The cytoplasm present in the cell has been shrinked. The shrink cell, that means the plasmolyzed cell, can be converted to deplasmolyzed cell by putting a water into it. Clear? So again hypotonic solution will yield. So that means the equal state is isotonic. If a cell shrinks, it's hypertonic. If a cell increases in size, is hypertonic. If a cell is equal, it's iso. If a cell is increasing in size, it is hypotonic. And if a cell is decreasing in size, is hypertonic solution. Now see active transport. Earlier I have said about the passive transport. Here active transport. That means what? Here movement of water molecules will go. That means by the help of energy. That means here ATP is used. That means what? Against the concentration it will go. In passive transport it was towards the concentration gradient. Here against the concentration gradient. So see, here are carrier proteins given. It helps to pass from outside to inside. What it helps? The solid particles to get inside the cell. Now two terminologies are given. One is endocytosis, another is exocytosis. Endo means what? Inside. So whenever any particles entering inside a cell, that is called endocytosis. Figure is given right hand side top. See, a molecule is getting inside a cell. Next one is exocytosis. You see what happens here? A molecule goes outside the cell. Figure is given right hand side, lower one. See, the molecules are going outside the cell. That's exocytosis. Now in this slide, we'll study about the functions of plasma membrane or cell membrane. So what is the function of cell membrane? It gives shape to a cell. Clear? It acts as osmotic barrier between the inner and the exterior of the cell. It facilitates diffusion osmosis in a cell. It helps as a hormone receptor in the target cells. It also promotes intake of liquid and solid food from the external medium by pinocytosis and phagocytosis. I have said pinocytosis means liquid intake, phagocytosis means engulfing of food. Clear? And last function is it protects the cellular living protoplasm from external environment. Clear? So here what we have studied: active transport, passive transport, diffusion, osmosis, hypotonic solution, isotonic solution, hypertonic solution. Clear? These are the terminologies. And fluid mosaic model. Study the diagram very nicely. It was given by Singer and Nicholson. So these are your homeworks. Thank you, student. This is all about the entire video lecture. Hope you have understood the video of cell membrane or plasma membrane. So stay at home, stay safe. I am Shomajit sir signing off for today, and again we'll meet in the next class. Thank you, students.